Hi friends, today we are going to discuss nuclear structure and functions. We will also illustrate the entry and exit of large molecules from the cell nucleus controlled by nuclear pore complexes. Let's first discuss nuclear envelope. The nuclear envelope is highly regulated membrane barrier that separates the nucleus from the cytoplasm in eukaryotic cells. It contains a large number of different proteins that have been implicated in chromatin organization and gene regulation. Although nuclear membrane enables complex levels of gene expression, it also poses challenge when it comes to cell division. The nuclear envelope is a hallmark of eukaryotic cells and is highly organized double membrane that encloses the nuclear genome. Early electron microscopy images revealed that the inner and outer nuclear membranes are continuous with the endoplasmic reticulum. Nuclear envelope formation has been studied in various cell-free systems, cell types and organisms. There are essentially two different ideas about how the nuclear envelope is formed. First, by the vesicle fusion, and second, by reshaping of endoplasmic reticulum into nuclear envelope sheets. Based on biochemical data and electron microscopy observations, it was initially proposed that nuclear envelope fragments into nuclear envelope vesicles. This idea is derived from cell-free systems, mainly of xenopus, starfish and Drosophila. High-resolution imaging in intact cells have revealed endoplasmic reticulum tubules contacting chromatin at early stages of nuclear envelope formation. According to this idea, the observed inhibition of nuclear assembly by fusion inhibitors and the role of senior proteins in nuclear envelope formation are likely to be an indirect effect of blocked endoplasmic reticulum reconstitution. Because nuclear envelope formation is also affected by endogenous levels of endoplasmic reticulum shaping reticulon proteins that slow nuclear envelope formation. These findings suggest a tug of war between the reticulons and their membrane curving activity and nuclear envelope proteins which promote membrane attachment and spreading around the chromatin. Now coming to our next topic, that is nuclear pore complex. The nucleus is bounded by a double membrane termed as nuclear envelope, which separates the nucleoplasm and genetic material from the surrounding cytoplasm. However, the genetic material in the nucleus is not totally isolated from the rest of cell. Studied throughout the nuclear envelope are portals called as nuclear pore complexes. Nuclear pore complex is highly selective bidirectional transporter for a tremendous range of cargoes. In electron microscope, the nuclear pore complex appears as a complex cylindrical structure with strong octagonal symmetry, measuring some 100 to 150 nanometers in diameter and 50 to 70 nanometers in thickness, depending on the organism. This overall appearance seems broadly conserved throughout all the eukaryotes. The two membranes of nuclear envelope, the outer and the inner membranes, join only in specialized, sharply curved piece of pore membrane that forms the grommet in the nuclear envelope within which NPC sits. Within each nuclear pore complex is a core structure containing 8 spokes surrounding a central tube. This central hole is about 30 nanometers in diameter and 50 nanometers in length, where the nucleoplasm connects to the cytoplasm and where the macromolecular exchange occurs. Peripheral filaments are attached to the core, filling the central hole as well as emanating into the nucleoplasm and cytoplasm. These filaments form a basket-like structure on the nuclear side of nuclear pore complex. One can envision the nuclear complex as being comprised of layers of interacting proteins, starting with the core structure, moving outwards through its peripheral filaments, and then to associating clouds of soluble transport factors, peripherally associating protein complexes in the nucleus and cytoplasm. These protein interactions can occur on radically different time scales. Some proteins form relatively permanent association with the core structure and are termed as nuclear pore complex components or nucleoporins, represented as NUPs. Other proteins associate transiently with the NPC, either constantly cycling on and off or attaching only at particular times in cell's life cycle. Nuclear pore complex is covered in binding sites for these transiently associating proteins because the nuclear pore complex is neither a motor nor an enzyme. The interactions provided by its binding sites wholly define the function of nuclear pore complex. The main body of nuclear pore complex comprises a surprisingly small number of 30 unique proteins. The structural proteins making up the bulk of the spokes and the rings give nuclear pore its shape and strength. These core proteins of nuclear pore complex also maintain the stability of nuclear envelope and facilitate bending of the pore membrane into inner and outer nuclear membranes. The most equatorial rings, termed the inner rings, are comprised of nucleoproteins 170 complex in yeasts and nucleoprotein 155 complex in vertebrates. The inner rings are sandwiched between the outer rings, which are comprised of nucleoprotein 84 complex in yeasts and nucleoprotein 107 complex in vertebrates. Together, these nucleoprotein complexes form a scaffold that hugs the curved surface of pore membrane and helps form the central tube through which macromolecular exchange occurs. Transmission electron microscopy sections cut normal to the nuclear envelope show continuity of nuclear envelope membrane with the endoplasmic reticulum and also reveal that the inner and outer nuclear membranes are fused to accommodate the pore structure. Field emission in-lens scanning electron microscopy visualizes the surface structure of nuclear pore complex and its eightfold radial symmetry wherein 8 filaments project into the cytoplasm at the nuclear surface, which contrasts with the characteristic nuclear pore basket structure and the nucleoplasmic phase of nuclear envelope. Now we will discuss about the functions of nuclear pore complex. All the transport between the nucleus and the cytoplasm passes through nuclear pore complex. The main function of nuclear pore complex is to control the passage of the molecules between the nucleus and the cytoplasm. A tight regulation of nucleocytoplasmic transport is essential for cell homeostasis. Consequently, alterations in nuclear pore complex membranes or the nuclear transport process have strong impact on cell growth and survival. Besides acting as the gatekeepers of the nucleus, it has become evident that nuclear pore complexes and nucleoporins are implicated in many other biological functions. Nuclear pore complexes have been shown to anchor and modulate the activity of simulating and dissimulating enzymes. Interestingly, nucleoprotein 358, the main component of cytoplasmic filaments, is an active E3 ligase in the simulation reaction. Last but not the least, several reports have associated nuclear pore complexes with chromatin organization and regulation of gene expression, especially in lower eukaryotes. Now we will briefly describe structure of nuclear lamina. Nuclear lamina is a structure near the inner nuclear membrane and peripheral chromatin. It is composed of lamins, which are also present in the nuclear interior and lamin associated proteins. The increasing number of the proteins that interact with lamins and the compound interactions between these proteins and chromatin associated proteins make nuclear lamina a highly complex but also very exciting structure. Nuclear lamina is an essential component of metazoan cells. It is involved in most nuclear activities including DNA replication, RNA transcription, nuclear and chromatin organization, cell regulation, cell development and differentiation, nuclear migration and apoptosis. Specific mutations in nuclear lamina genes cause a wide range of heritable human diseases. These diseases include limb girdle muscular dystrophy, dilated cardiomyopathy with conduction system disease, familial partial lipodystrophy, and autosomal recessive axonal neuropathy. Now coming to our next topic, in which we will illustrate transport across the nuclear envelope. The entry and the exit of large molecules from the cell nucleus is tightly controlled by nuclear pore complexes. Although small molecules can enter the nucleus without regulation, macromolecules such as RNA and proteins require association with caryophorins called importins to enter the nucleus and exportins to exit. Proteins that must be imported to the nucleus from the cytoplasm carry nuclear localization signals abbreviated as NLS that are bounded by importins. NLS is a sequence of amino acids that act as tag. They are diverse in their composition and most commonly hydrophilic, although hydrophobic sequences have also been documented. Proteins transferred RNA and assembled ribosomal subunits are exported from the nucleus due to the association with exportins, which bind signaling sequences called nuclear export signals abbreviated as NES. The ability of both importins and exportins to transport their cargo is regulated by an enzyme GTPase RAN. GTPases are the enzymes that bind to the molecule called guanosine triphosphate, abbreviated as GTP, which they then hydrolyze to create guanosine diphosphate and release energy. RAN is in different conformation depending on whether it is bound to GTP or GDP. In its GTP bound state, RAN is capable of binding caryophorins, importins and exportins. Importins release cargo upon binding to RAN GTP, while exportins must bind RAN GTP to form ternary complexes with their export cargo. The dominant nucleotide binding state of RAN depends on whether it is located in the nucleus or the cytoplasm. Now let us first discuss nuclear import. Important proteins bind their cargo in cytoplasm, after which they are able to interact with the nuclear pore complex and pass through its channel. Once inside the nucleus, interaction with the RAN GTP causes conformational changes in the importin that causes it to dissociate from its cargo. The resulting complex of importin and RAN GTP then translocates to the cytoplasm, where a protein called RAN binding protein RNBP separates RN GTP from importin. Separation allows access to GTPase activating protein gap that binds the RAN GTP and induces the hydrolysis of GTP to GDP. The RAN GDP produced from this process now binds the nuclear transport factor abbreviated as NUTF2, which returns to the nucleoplasm. Now in the nucleus, RAN GDP interacts with guanine nucleotide exchange factor abbreviated as GEF, which replaces the GDP with GTP, resulting again in RAN GTP and beginning the cycle anew. Now coming to next, that is nuclear export. Nuclear export roughly reverses the import process. In the nucleus, the exportin binds the cargo and RAN GTP diffuses through the pore to the cytoplasm, where the complex dissociates. RAN GTP binds gap and hydrolyzes GTP. The resulting RAN GTP complex is restored to the nucleus, where it exchanges its bond ligand for GTP. Hence, whereas importins depend on RAN GTP to dissociate from their cargo, exportins require RAN GTP in their order to bind to their cargo. A specialized mRNA exporter protein moves mature mRNA to the cytoplasm after post-transcriptional modification is complete. This translocation process is actively dependent on RNA protein, although the specific mechanism is not yet well understood. Some particularly commonly transcribed genes are physically located near nuclear pores to facilitate the translocation process. tRNA export is also dependent on the various modifications it undergoes, thus preventing the export of improperly functioning tRNA. This quality control mechanism is important due to tRNA's central role in translation, where it is involved in adding the amino acids to a growing peptide chain. tRNA exporter in vertebrates is called exportin T. Exportin T binds directly to tRNA cargo in the nucleus, and a process is promoted by a presence of RAN-GTP. Mutations that affect tRNA's structure inhibit its ability to bind to exportin T and consequentially to be exported, providing the cell with another quality control step. As described above, once the complex has crossed the envelope, it dissociates and releases the tRNA cargo into the cytosol. Protein shuttling is also included in its transport. Many proteins are known to have both NES and NLS and thus shuttle constantly between nucleus and the cytosol. In certain cases, one of these steps, that is nuclear import or nuclear export, is regulated, often by post-translational modifications. Our next topic is chromatin and its molecular organization. Chromatin is the assemble of the nuclear DNA and all associated proteins and RNA molecules. The chromatin proteome is highly complex. It consists of histones that carry a wide range of post-translational modifications and hundreds of other proteins that can interact with DNA, with modified histones and with each other. The fundamental unit of the chromatin is termed as nucleosome, which is composed of DNA and histone proteins. This structure provides first level of compaction of DNA into the nucleus. Nucleosomes are regularly spaced along the genome to form nucleofilament, which can adopt a higher level of compaction, ultimately resulting in the highly condensed metaphase chromosome. Within an interphase nucleus, chromatin is organized into functional territories. Chromatin has been divided into two types, euchromatin and heterochromatin. Heterochromatin was defined as the structure that does not alter in its condensation throughout the cell cycle, whereas euchromatin is decondensed during the interphase. Heterochromatin is localized principally on periphery of the nucleus and euchromatin in the interior of the nucleoplasm. We can also distinguish heterochromatin into constitutive heterochromatin and facultative heterochromatin. Constitutive heterochromatin containing few genes and formed principally of repetitive sequences located in a large regions coincident with centromeres and telomeres. Facultative heterochromatin is composed of transcriptionally active regions that can adopt the structural and functional characteristics of heterochromatin, such as inactive X chromosomes of mammals. Now coming to our next important topic, that is the nucleosome. The nucleosome is the fundamental unit of chromatin. It is composed of a core particle and a linker region or internucleosomal region that joins adjacent core particles. The core particle is highly conserved between the species and is composed of 146 base pairs of DNA wrapped 1.7 turns around a protein octamer of each of the core histones H3, H4, H2A and H2B. The length of the linker region, however, varies between species and cell type. It is within this region that variable linker histones are incorporated. Therefore, the total length of DNA in nucleosome can vary with species from 160 to 241 base pairs. Analysis revealed firstly the distortion of the DNA wound around the histone octamer, and secondly that the histone to DNA and histone to histone interactions through their histone fold domain formed configuration reminiscent of handshake. Let's first discuss histone proteins. Core histones. The core histones include H3, H4, H2A and H2B. These are small basic proteins highly conserved in evolution. The most conserved region of these histones is their central domain, structurally composed of histone fold domain, consisting of 3 helices separated by 2 loop regions. In contrast, the N-terminal tails of each core histone is more variable and unstructured. The tails are particularly rich in lysine and arginine residues, making them extremely basic. This region is the site of numerous post-translational modifications that are proposed to modify its charge and thereby alter DNA accessibility and protein-to-protein interaction with nucleosome. It is significant to note that other proteins that interact with the DNA also contain the histone fold domain. Now, what is the linker histones? Linker histones associate with the linker region of DNA between 2 nucleosome cores, and unlike the core histones, they are not well conserved between the species. In higher eukaryotes, they are composed of 3 domains: a globular, a non-polar central domain essential for interaction with DNA, and 2 non-structured N and C terminal tails that are highly basic and proposed to be the site of post-translational modifications. The linker histones have a role in spacing nucleosomes and can modulate higher order compaction by providing an interaction region between adjacent nucleosomes. Now I will conclude my lecture with my last topic on rRNA processing. Ribosomal RNAs are the most abundant and universal non-coding RNAs in living organisms. In eukaryotes, 3 of the 4 ribosomal RNAs forming the 40S and 60S subunits are bound by a long polycistronic pre-ribosomal RNA. A complex sequence of processing steps is required to gradually release the mature RNAs from this precursor, concomitant with the assembly of 79 ribosomal proteins. A large set of trans-acting factors chaperone this process, including small nucleolar ribonucleoparticles. While yeast has been gold standard for studying the molecular basis of this process, recent technical advances have allowed to further define the mechanisms of ribosome biogenesis in animals and plants. This renewed interest for long-lasting questions has been fueled by the association of several genetic diseases with the mutations in genes encoding both ribosomal proteins and ribosome biogenesis factors, and by perspective of new anti-cancer treatments targeting the mechanism of ribosome synthesis. A consensus scheme of pre-ribosomal RNA maturation is emerging from the studies in various kinds of eukaryotic organisms. However, major differences between mammalian and yeast pre-ribosomal RNA processing have recently come to light. The genes for rRNA, like those of tRNA, can be present in multiple copies, and the numbers vary among the species. All copies of the rRNA genes in a species are identical or nearly identical. In bacteria, rRNA genes are dispersed, but in eukaryotic cells, they are clustered with the genes arrays in tandem one after another. Eukaryotic cells possess 2 types of rRNA genes: a large gene that encodes 18S rRNA, 28S rRNA and 5.8S rRNA, and a small gene that encodes 5S rRNA. All the 3 bacterial rRNAs, 23S rRNA, 16S rRNA and 5S rRNA, are encoded by a single type of a gene. Ribosomal RNA is processed in both bacterial and eukaryotic cells. In E. coli, the immediate product of the transcription is 30S rRNA precursor. This 30S precursor is methylated in several places and then cleaved and trimmed to produce 16S rRNA, 23S rRNA and 5S rRNA along with one or more tRNAs. Eukaryotic RNAs undergo similar processing. Small nucleolar RNAs help to cleave and modify eukaryotic rRNAs and assemble the processed rRNAs into mature ribosomes. Like small nuclear RNAs taking part in pre-mRNA splicing, small nucleolar RNAs associate with the proteins to form ribonucleoprotein particles. These ribonuclear particles have extensive complementary to the rRNA sequences in which modification takes place. Interestingly, some nucleolar RNAs are encoded by sequences in the introns of other protein encoding genes. The processing of rRNA and the ribosomal assembly in the eukaryotes takes place in nucleolus. With this, I conclude my topic. Thanks for your patience.